Well, I'm going to talk a little bit about the role of carotenoids in the eye and give you a review of what we've done in part. And first part of the review is where we did the work. And unfortunately, it wasn't on South Beach in the Art Deco district, but we do live a little bit down the road from this nice area. And so I thought I'd bring a picture and show you a little bit of our local scenery. The one thing I want to start off with is that in the last conference, we had a wonderful set of speakers who all agreed to contribute to a volume, and John Nolan and I have put this together. Unfortunately, it has not yet reached full printing, but I wanted to point out to you that it's available at a 20% discount. So if you were going to purchase it, you know, there's a code that you could get it reduced price. And Alan, are you in the back? Please stand up, because I want to acknowledge that the book is dedicated to Alan and his support for research into the carotenoids and these conferences. And so it will be coming out in October, and we're very grateful for his support to these conferences and in research for macular pigments in the eye. So without further ado, let me tell you about the macular pigments. And if you weren't here yesterday, you didn't see these, but if you were here, I'm sure you did see these photographs. They are very famous. The now famous photograph that appeared in Martha Nuringer and Malinois' paper in 1980 of the fundus of the macaque retina showing the carotenoid pigment. And then I had hoped we would have Max Snodderly here, but I understand that he will not be. But he took these wonderful micrographs in 1984, and you can see the macular pigment here. So we see the macular pigment is localized in the fovea, and we see furthermore that it has a very unique distribution through the depth of the retina. It is in the inner regions, in the henofiber regions of the macula, and we see very clearly how well it absorbs blue light. And yesterday, I'm sure we were hearing that the absorption can be as much as 40–50% of the total incoming blue light. This is attenuating a lot of the light and reducing the chances of photooxidative processes dramatically in the retina. So the notable things that we learned from those photographs, and remember, those in the early 80s, were that the amounts are localized, they're in the inner layers, and the distribution is not uniform. So we find that in the outer photoreceptor layers, we only found that in the very central fovea that there were significant amounts of carotenoids. And that's about the time when my colleague and I, Richard Bowen, joined into this research and began to study. And so there are several questions that we had clearly at that time, and we have really still today. There are many, many questions. So I'm going to leave you with more questions than I'm going to answer. And I think that that's very good, because that means that there's a lot more for us to do in the upcoming years. The goals Paul set for us 2 years ago are not fully realized. We've made some strides, but it's going to be a little bit longer before we know everything. Where is the macular pigment? In what cell types are they found? Where are they found within those cells? What role can the pigment play in each location that it is found? And do the different carotenoids have different functions? Are they differentially distributed across the retina? Some of those answers began to appear a while back, but those answers aren't fully available to us. I want to remind you, and we're going to look at this again and again throughout the conference, of the structure of the macula. We all know that the macula is at the focal point at the back of the eye. The structure is an inverted retina structure with the cones directed to the outside and rods, the retinal pigment epithelium, and much of the action in the retina is down here. the processing occurs here, and then, of course, that leads out to the optic nerve. And, of course, the macular pigment is in that zone. So what should we be thinking about? Well, we've been concerned with the pathology, the ways in which there might be disease processes in the eye and AMD. And so one of the concerns that we have all had are what are the species causing these. And the oxidative species are these radicals. They differ a lot in their properties. Some are extremely reactive, hydroxyl and alkoxyl radicals. Singlet oxygen, their lifetimes are very short. They react in very, very short periods. And then others are much longer lived. Nitric oxide, which we now know is a very, very important signaling molecule, peroxyl radicals, superoxide, and hydrogen peroxide. These also are signaling molecules, and their roles are becoming better formed. George, you did a great job in showing us how light generates singlet oxygen, so I won't review that, but I will emphasize that there are a number of species that are in the retina that could very easily act as sensitizers. Heme, tryptophan, vitamin A. So there are a lot of molecules in the eye that can do that. There is clearly a lot of oxygen in the retina. It's a very active aerobic tissue. And clearly, without light, the eye doesn't function. So all the components for generating this very reactive and dangerous species are there. Peroxide is generated by mitochondrial oxidase dysfunction. Normally, we don't expect the mitochondria to produce and release peroxide, but it does. There are some enzymes that do this. Superoxide can generate this from the superoxide ion that is formed also in mitochondrial dysfunction. Superoxide is also produced in white blood cells as a natural course. Nitric oxide I've put in here as an example of a radical that is ubiquitously found in the eye and in other tissues and can have reactive consequences. So just to keep us in tune, all radicals aren't bad all the time. And look at this, plants. We regulate metabolism, growth, seed germination, their immune response. Even plants have immune response. Cell growth, differentiation. Animals, vaso and pulmonary dilation and constriction. Same molecule, constriction on one hand, vasodilation on the other hand. Detoxification of xenobiotics by peroxide, catabolism. Nitric oxide controls cell growth. These are very important molecules. They appear over short time spans and then disappear. They're very good as signaling agents. Many of them, because they don't live long, won't travel very far from their source, so the signal is in a local region. They're very good for triggering differentiation and making sure that what happens right where they are formed. So on the other hand, if we form too many radicals, we're going to have a case where structures, molecules that are sensitive to these radicals react and are degraded. And so we do need antioxidants, both intracellularly and extracellularly, both within the cytosol, the aqueous environment, and in the membranes. And George was talking to us about the properties of carotenoids, and carotenoids are, by and large, lipophilics. And so we have abundant non-enzymatic antioxidants in the water section of our tissues: thioredoxin, vitamin C, melatonin, uric acid, lipoic acid, many of the thiols, glutathione. They're all very good antioxidants. So the aqueous portion of the cell is richly covered. It really the membranes and the lipid fractions where we need this better coverage for protection against radicals. And carotenoids, as we heard George tell us, love these lipid environments. And different carotenoids take up different sites within the membranes and lipophilic structures themselves. Tocopherols are also found there, and so are some other important species. So bring you back to these radicals one more time. lifetimes tell us about the distances these molecules can move. So hydroxyl radical is only going to move 40 to 100 angstroms from where it's formed. It's so hot, it's like a little hand grenade. It's going to react right where it's formed. Singlet oxygen, it's about 100 times longer lived. It's going to be able to move on the order of maybe up to 100 micrometers in very non-viscous media. But remember, cells aren't non-viscous media. They're jelly-like, and so these estimates are very crude, and this may be a vast overestimate in the cellular system. These three down at the bottom now I've highlighted and made a different color because their lifetimes are quite long. These species, once they're formed, can move around. They have plenty of time to get from various places. Hydrogen peroxide is neutral. It can actually, although it's not well solubilized by membranes, move through membranes and into membranes and across them. So we have a lot of opportunity for these molecules to move around and do various kinds of things. So having said all those things about the radicals, let's just talk one more little bit about what goes on. Transcription processes of many regulatory proteins are affected by radicals. So we have NF-kappa B, TIMP, COX, NOx, AP1, SOD, catalase, both species that promote oxidative processes and those that control oxidative processes. Their transcription is related to radical production. So if carotenoids influence radicals, if carotenoids interrupt the numbers of radicals formed, then it's going to alter these transcription processes as well. So here are our players. George introduced you. You need no further explanation of these two folks. Richard and I were astounded when we found that there were two components in our first HPLC fraction of the macula back in, I think that was about 1983. It took us a little while longer to actually map the carotenoids, but here you see what you would have expected from that very first photograph. The carotenoids are concentrated in the macula. What you would not have expected, what you would have anticipated would be that the ratio of lutein to zeaxanthin would have remained constant across the retina. Because the dietary supply has a ratio, you would have expected that the carotenoid ratio across the retina would remain the same as that of the diet. And no, it's not. The ratio in the peripheral regions of the retina is about 2.5 lutein to zeaxanthin. And remember what George told you about 10-to-1 lutein to zeaxanthin in the diet. So this is quite a difference from the diet even. Already we're seeing some differences from the diet. But as we move into this region of the central macula, we find that the ratio has a dramatic shift. And the ratio drops below 1, in fact, about 1 to 2. Let me put it this way, about 4-to-1 zeaxanthin to lutein, a shift the other way. And so we have some things going on. And George was talking to you about mesozeaxanthin, and mesozeaxanthin was suggested by us to form from lutein. And so we looked at this in the early 90s, and we looked at the ratio of mesozeaxanthin, and here is the ratio of mesozeaxanthin. Unfortunately, we don't have the resolution in our HPLC data. We only have 3 points leading up here. It probably peaks a lot higher in the very central part of the macula, and so this tells us that mesozeaxanthin is being formed here in the central part of the retina. This is the mesozeaxanthin-zeaxanthin ratio, and so we see that changing most dramatically here in the center, which tells us that this is occurring in one part of the retina more than another. It's not occurring, it would not seem to be occurring as a metabolism in the liver and then transported, because again, then we would have thought we would have a uniform distribution, which we do not. So Liz Johnson helped us out at this point. She did this wonderful study with Martha Nuringer and others here, and showed that if you fed macaques lutein, you would see mesozeaxanthin in the retina. If you fed them RR zeaxanthin, you would not. And so they did not see any in the blood. They saw it only in the eye. And that pretty well convinced us that we had gone on the right direction, that in the eye, lutein is being converted to meso-zeaxanthin. And as George pointed out, the stereochemistry here and the stereochemistry here suggested that was the case. But I'm going to tell you one more thing that most people seem to overlook in one of our papers, and that is that although meso-zeaxanthin is the principal secondary metabolite in the retina, SS-zeaxanthin is there. It's not absent. So there is another isomer present, and that's a very interesting thing. How does this conversion occur? Well, your guess is good. There are a couple of ways that the literature suggests. One is oxidation of the hydroxyl group, which would be loss of chirality, and then re-reduction. Re-reduction could be with a chiral preference. Enzymes do have chiral preferences. Evidence to support this kind of mechanism is that there are oxidation-reduction mechanisms of conversion to xanthophils in other organisms, not necessarily this specific one. Reductive conversion of canthaxanthin, which was a tanning agent very popular in the 70s and 80s and accumulates in the retina, to isozeaxanthin is known. So reduction of keto groups to hydroxyl groups is known. The alternative mechanism is just simply the migration of the double bond in lutein from the position here, which is the 4-5 position, to the is that right? 4-5 to the 5-6 position, yes. And so there are also enzymes that shift double bonds, so that could be a mechanism by which that occurs. I want to shift a little bit. I want to go back now and talk a little bit about distribution, and I want to talk about maturation of the eye. Because the macular pigment doesn't just happen to be in adults, it happens to be in children. But when you look carefully, you find that children are a little different than adults. So these are some macaque retinas, and I want to thank Martha for the tissue samples that she sent to us. And these are in 2 millimeter, 4 millimeter, and 8 millimeter concentric regions around the fovea. And you see in this region, the amount of total zeaxanthin to lutein gets very high in the central part of the eye and then drops off. So there's a lot of zeaxanthin in the central part of the retina. And these are adult animals. Now, we did look at macular carotenoids in the early years of life in the human eye in our 1988 paper. And you see those data here. The amounts of carotenoid are usually very low at birth, but they rise very quickly. Moms love to feed their children these green peas things, you know, and they feed them other things that are rich in lutein and zeaxanthin. And so right away we start to pigment the eye. But interestingly enough, the ratio of lutein to zeaxanthin between 0 and, well, 3 years seems to be greater than 1. So lutein dominates in the fovea until about 3 years in humans. And then something happens. It changes. Well, what is that something? Well, we know now. That's the conversion of lutein into meso-zeaxanthin. You see this metabolism kicking in at about that point. And in the first 4 years of life, there is a tremendous amount of development in the retina as well. So that accompanies the structural and metabolic changes in the retina. So here are these sections of the tissue that we're looking at: a 2 millimeter punch, a ring around at 4, and then a ring at 8 millimeters. And now I'm going to show you some data from young macaque retinas. Now, these are not all shown in the same way. This is only up to 2 years. These go into the adult years, and I've extended the line a little further here. But I'm only going to look at this for the most part, this early part of the eye. So we have lines that we fit to these data. They're not a lot of data, I admit, and so the fits are not as good as I would like, but we can use these, and I have this for the 2 millimeter, the 4 millimeter, and the 8 millimeter region. So if we then take these lines, and then which are a function of age and concentration, we can then make a picture of what the macula concentrations ought to look like. And so at about 0 years, birth, we should see a little bit of lutein in the eye. That's what this looks like. We don't go to a 0 intercept. And these are linear fits, so this increases with time up to about 2.5 years. And we're getting to a value that's somewhere around 2 nanograms per square millimeter. Let's look at the total zeaxanthin. And these are the same scale. So I think that that's interesting. Look at that. Zeaxanthin also starts out present in the eye, but at smaller amounts than lutein, but it rockets up. The amount goes up much, much faster. That's exciting. There's a difference in those rates. Let's break RR zeaxanthin down into its 2 components, and we see that the RR and the RS, I'm not sure that this is a significant difference here because these are the results of linear fits. So they start out very low and then increase. Meso seems to increase a little bit faster based on this limited data set that we have. So you can look at the profiles and now see that in the early developmental period in the macaque, over about the first 2 years, you've got this rapid change in the amounts of carotenoids. We can look at the ratios from those numbers also, and we see that the ratio where mesozeaxanthin and lutein reach unity happens to occur at about 1.5 years. Interestingly, the time when lutein and the total zeaxanthin reach unity is, maybe 6 months, okay, half a year or thereabouts. So much faster. The zeaxanthin level is surging up. And this is a little bit like George was saying. You've got that extended conjugated system. The zeaxanthin may be a better quencher of singlet oxygen. And George talked to you about the structural difference of the carotenoids, and I'm just going to remind you that, as he said, the epsilon ring of lutein is very contorted. It's distorted by the fact that it's got a tetrahedral carbon linking to the polyene system, whereas the beta ring that is in zeaxanthin is very linear. And this makes a wonderful high-fidelity way for molecules to recognize and distinguish the lutein and zeaxanthin. Now, mesozeaxanthin, the hydroxyl group would point down this way instead of up that way. So that's the distinction that you would see here. So I'm going to try and finish off and remind you that carotenoids can intercept reactive species. They are proven in many systems to protect against light and oxygen, generation of singlet oxygen and other reactive species. But I want to remind you carotenoids are also cleaved to form apocarotenals, both by simple reactions and by enzymes. There are 2 enzymes, beta-carotene dioxidase or carotenoid dioxidase 1 and 2. The 1 does not actually cleave the zeaxanthin molecule. It cleaves beta-carotene to form retinal. But in insects, there is an enzyme that does this. So I've left it in that form, not in humans, I think. But this enzyme cleaves the carotenoids here. And so these 2 molecules present themselves as metabolites that may well be signaling molecules. These may be able to react with, once they're formed, react with proteins and therefore modulate transcription processes. To my knowledge, carotenoids as whole species don't enter the nucleus. And that's a question that we should perhaps look at, is which species might be getting into the nucleus to modulate transcription events. So again, if we're going to modulate these sorts of species, we have to modulate them where they're formed or where they're reaching. Where are the carotenoids? Are they near the formation of these species? Because that's where they'll have to be if they're going to get these. Or are they in some place where they can be involved with reacting with these species before they migrate? Carotenoids will, the lutein and zeaxanthin will be found in lipid membranes. So we have a lot of lipid membranes in the rod outer segments and the cone outer segments. They fit there almost perfectly. The dimensions are a good match. Paul has taken these wonderful micrographs of two proteins, STARD3. I'm not going to even try and give its full name. It's steroidal acute regulatory domain. It's a long name. These proteins are normally cholesterol transporters, but they also are very good transporters. It turns out this particular enzyme or protein is very good at transporting lutein, and it binds lutein very strongly. If we expand this is a region, it's shown in Paul's very nice paper. The red shows where this protein lies, and it is a lutein binding protein. What we're looking at is the protein. We don't know if we're looking at all the lutein in the eye, but we're looking at where that lutein binding protein is. And so we also see it in the RPE there. That's very interesting, I think. And here's another protein which Paul has identified for us. I'll stick with the GSTP1. Boy, look at it light up red there. We don't see the RPE here. I think the micrograph, they lost the RPE, but boy, it lights up. And when I look at that, Paul, I looked at it real closely. It looks to me like they're little organelles that are specifically staining. It might be mitochondria. And so, you know, we've been speculating that reactive species might be coming from the mitochondria. So if you're staining the mitochondria and these proteins are being formed in the mitochondria, that might be a very clear mechanism for protecting the retina from mitochondrial dysfunction. So that's kind of interesting as well. So, sum up. Carotenoid distributions are biologically controlled by solubility, by the specific binding proteins, and they're structure dependent. And there's a lot of fidelity in identifying these two structures. These two proteins really discriminate between lutein and zeaxanthin. The carotenoids are reactive with reactive oxygen species. They can quench singlet oxygen directly. They're good membrane protectors. There are a lot of membranes in the eye. They can protect those unsaturated fatty acids that are so abundant in the eye. Carotenoids are enzymatically cleaved to produce apocarotenoids. These are showing up, at least lycopene metabolites. There's a lot of evidence showing that they're controlling transcription events. And so this seems like a new plausible direction. I want to thank my colleague Richard Bone for many years. I want to thank Martha for those wonderful tissue samples, my student Issa Sal. I want to thank all the people who have helped fund our research over the many years. And I want to thank you all for your patience and listening to me and the organizers, Dr. Howard, John, Stephen. Thank you very much.
Now, are there any questions? I see Toss has one question for you there.
Thank you for your talk. So you showed us that zeaxanthin is preferential in the very center. So I guess there must be a functional reason for it. But you also showed that in humans, it does only occur after 3 years of age, and in macaques maybe somewhat earlier. So is this only because only at that age you are able to get meso-zeaxanthin there because it's a transversion process, or is there a functional reason that it occurs after 3 years of birth? Can you comment on this?
I can speculate.
Yeah,
okay.
Please do.
Because clearly, what we need to do is to have a retina which maybe we grow ex vivo, and we do real-time PCR to see the transcription of these proteins and understand that, lacking that possibility at the moment. what we just have to say is that the retina in the human develops over this 4 years of life from birth. It's a slow process. The human retina is not born fully developed. The cones are not elongated. There is no foveal depression. And it seems that it's only at the same time as the retina matures that you see this fully mature distribution of carotenoids as well. Does that mean that very young eyes don't benefit from carotenoids? I don't think that that is what one would conclude necessarily. It may be that until you develop the full retina, you haven't had time to accumulate and transport in from a normal diet enough carotenoid and do enough metabolism to have this normal distribution.
John, we talk about the conversion of lutein to meso-zeaxanthin, and George discussed nicely that it's likely that it happens from lutein. How natural is that process? Okay, my mic. How natural is that process, do you think? I suppose my question is, do you think everybody can do it? Do you think it's something that happens easily? Is it all enzymatic? Is there a mixture of factors?
This is certainly, you know, a big question, because we know that there are many mutations that exist in enzymatic systems, and they're responsible for many pathologies. You know, the thalassemias, cystic fibrosis, etc. And so it seems reasonable that the enzymes that are responsible for meso-zeaxanthin formation would be susceptible to the same problems of mutation, and there ought to be polymorphisms. And if that's the case, then probably some of us, maybe in this room, do not have very robust enzyme systems or even capable enzyme systems that can make meso-zeaxanthin. And so if that were the case, it would be important that we supplement in order to get to normal zeaxanthin levels.
Yeah, just to comment on that and add to that, I certainly in looking at autopsy eyes, and I think John would agree with this, there's always some meso-zeaxanthin in it. I've never seen a retina that didn't have meso-zeaxanthin when you specifically look for it. We can speculate on enzymes. We don't know the enzyme yet. I'm still trying to work on that, and I'll discuss some of that in my talk. So we have to look at that. I think the other thing that I'll discuss in my talk too is you have to look at the oxidation and perhaps recovery of these oxidation products that you talked about before. That may be where some of the meso-zeaxanthin is coming from, and that may explain why you see this delay in children, because they're not at birth. They're not exposed to. They haven't been exposed to light, and there is a couple of years that it takes of light exposure that occurs that may be allowing some of the meso-zeaxanthin to build up from oxidation and other stresses that are finally accumulating in the eye.
Mm-hmm. Mm-hmm. Okay, well, if there are no further questions, we'll move on to our next speaker.
I just have one quick question, if you don't mind. Thank you for a lovely talk, John. That's really, really interesting. I just have one question concerning the distribution of meso-zeaxanthin to zeaxanthin in the chart that you showed, and also considering the fact that it's the same protein that it seems to bind. Do you have any idea why there is a differing ratio, or what produces that?
I'm not sure I understood fully what you're trying to ask.
Well, you know the chart where you showed the meso-zeaxanthin to zeaxanthin ratio in the macula, and then later on you discussed the GSTP1 protein, that it binds both zeaxanthin and meso-zeaxanthin. I'm just wondering why there's a differing ratio, that there's a greater amount of meso centrally than in the, I suppose, peripheral region of the zeaxanthin area. Do you know what I mean?
Well, I think Paul would be better able to answer this, but I think the GSTP1, where in the micrograph we saw, was at the very, very center of the fovea. I think if you went out further, you might see that protein has a different abundance, and so you might find that it's not accumulating the zeaxanthins there. I think that is not necessarily the enzyme that's responsible for meso-zeaxanthin formation. It's just a zeaxanthin binder.